Welcome all in my lecture. Today we will learn how to search in the RCSB database. To search for specific information in the RCSB protein data bank, you can use their website's search functionality. RCSB PDB is a comprehensive resource for structural biology and contains a vast amount of data on macromolecules like proteins and nucleic acids. Here's a general guide on how to perform a database search on RCSB PDB. Access the RCSB PDB website, open your web browser and go to the RCSB PDB website at https://www.rcsb.org. Use the search bar. On the main page, you will find a prominent search bar on the top. You can use this search bar to search for various types of information. Keyword search. You can enter keywords related to the protein, structure or any other information you are looking for. For example, you can enter the name of protein, a PDB ID or a biological function. Advanced search. Click on the advanced link next to the search bar to access more specific search options. This allows you to refine your search by criteria like structure, resolution, experimental method, organism or more. Enter your search query. Enter your search query into the search bar and press enter or click the search icon. Browse search results. The search results page will display a list of entries that match your queries. Each entry represents a specific protein structure or related data. Filter the refined result. How you can filter your refined result? You can further filter the refine your results using the options on the left sidebar. You can filter by experimental methods, organism, publication date and many more. Explore specific entries. Click on a search result to access detailed information about protein structure including 3D visualizations, metadata and related publications. Download data. How we can download the data? If you want to download the structural data for a particular entry, you can usually find download options on the entry page. RCSB PDB provides various formats of downloading structural data. Citing data. If you plan to use the data in your search or publication, make sure to cite the relevant PDB entries according to the provided citation information. Remember that RCSB PDB website continually updates and improves its search and analysis tools, so the exact user interface may change over time. Always refer to the website's documentation and help resources for the most up-to-date information on conducting searches and accessing structural data. Now my colleague Josna will explain you the searching through RCSB database in detail.
Hello everyone. Today's lecture is on protein data bank, that is PDB, a very important resource for protein. It is the single worldwide archive of structural data of biological macromolecules. Structure in PDB have wide application. They can be used for various studies including identification of new protein structure via in silico approaches or can be used for protein nucleic acid interaction studies. Here you can get information about protein such as structure of protein, its function and different binding ligand and so many information about the protein. In these presentation, we have also included briefings about various file format available in PDB with special emphasis on PDB file format. PDB is a worldwide central repository of three-dimensional structure data of biological macromolecule such as protein, nucleic acid and other biological macromolecule. It was established at Brookhaven National Labs in 1971 as an archive of biological macromolecular crystal structure. Since October 1998, The PDB database has been managed by Research Collaboratory for Structural Bioinformatics, which is a consortium consisting of Rutgers, the State University of New Jersey. In 2003, the formation of worldwide PDB, the PDB became an international organization. In 2006, the Biological Magnetic Resonance Data Bank joined the worldwide PDB. Structural files in PDB are deposited by international scientific community and maintained by the RCSB PDB staff. Approximately 50 to 100 new structures are deposited every week. They are annotated by RCSB and released upon the depositor's specification. The PDB database is updated weekly. As of 30th August 2023, nearly 2,24,447 structures have been deposited in the PDB. A variety of information associated with each structure is available, including sequence detail, atomic coordinates, crystallization condition, 3D structure of protein, their experimental methods, geometric data, structural factors, 3D images, and a variety of links to other resources. PDB data is freely available worldwide. Information on structures can be retrieved from the main PDB website, that is www.pdb.org. Structure files can also be obtained through the main FTP site at ftp.rcsb.org. Presently, PDB is under the purview of the Worldwide Protein Data Bank, a network for 4 organizations, that is Protein Data Bank in Europe, Research Collaboratory for Structural Bioinformatics Protein Data Bank USA, Protein Data Bank Japan, Biological Magnetic Resonance Data Bank USA, whose mission is to maintain a single protein data bank archive of macromolecular structural data that is freely and publicly available to the global community. This is the homepage of Protein Data Bank in Europe. Protein Data Bank in Europe provides rich information about all PDB entry. It has multiple search and browse options. It provides various advanced search such as PDB-PISA, that is Protein Interface Structures and Assemblies. PDB-PISA is an interactive tool for the exploration of macromolecular interface. With PDB-PISA, you can retrieve pre-calculated results of whole PDB archive. You can calculate results interactively for structures uploaded as a PDB or MMCIF file. Next service is PDB Fold. It provides pairwise comparison and 3D alignment of protein structure. It provides multiple comparison and 3D alignment of protein structures. It also provides examination of protein structure for similarity with the whole PDB archive or scope archive. Next service of PDB Europe is PDB Chem. It is the dictionary of chemical components such as ligands, small molecules and monomers referred to in the PDB entries and maintained by Worldwide PDB. It provides comprehensive search facilities for finding a particular component or determining component in structure entries or vice versa. The next services in PDB Europe is PDB Motif. It is an extremely fast and powerful search tool that facilitates exploration of the protein data bank by combining protein sequence, chemical structure and 3D data in a single search. PDB in Europe Advanced Search and many other services also available in PDB in Europe data bank. This is the home page of RCSB PDB. It provides simple and advanced searching for macromolecules and ligands. It is specialized visualization tools. In RCSB PDB, sequence structure comparison, also there is a RCSB PDB mobile site is also available. It also provides molecule of the month and other educational resources at PDB 101. This is the home page of Protein Data Bank in Japan. The Protein Data Bank in Japan is a member of Worldwide Protein Data Bank. It accepts and process the deposited data of experimentally determined micromolecular structures. While maintaining the archive in collaboration with other worldwide PDB partner, PDB Data Bank in Japan also provides a wide range of services and tools for analyzing structures and functions of protein. It supports browsing in multiple languages such as Japanese, Chinese and Korean. Till now, PDB Data Bank Japan consists of nearly 2,09,389 structures. This is the home page of Biological Magnetic Resonance Data Bank. The Biological Magnetic Resonance Data Bank is a repository for data for NMR spectroscopy on protein, peptides, nucleic acid and other biomolecules. The file format initially used by the PDB was called the PDB file format. The original format was restricted by the width of computer punch card to 80 characters per line. Around 1996, the macromolecular crystallographic information file format MMCIF, which is an extension of CIF format, was phased in. MMCIF became the standard format for PDB archive in 2014. In 2019, the worldwide PDB announced that deposition for crystallographic method would only be accepted in MMCIF format. An XML version of PDB called PDBML was described in 2005. The structure file can be downloaded in any of these three formats, though an increasing number of structures do not fit the legacy of PDB format. Individual files are easily downloaded in graphic package from internet URL. The Protein Data Bank is a repository of atomic coordinates and other information describing protein and other important biological macromolecules. Structural biologists use methods such as X-ray crystallography, NMR spectroscopy and cryo-electron microscopy to determine the location of each atoms relative to each other in the molecule. They then deposited this information and publicly release into the archive by the worldwide PDB. RCSB PDB website allows you to search and explore the information under PDB header, including information on experimental method, chemistry and biology of protein. Once you have found the PDB entries which you are interested in, you may use visualization program to allow you to read PDB file in ResMol, which display the protein structure on your computer. Download the information and create custom picture of it. One can search for their protein of interest by using the search bar in RCSB PDB website. PDB can be searched by providing three methods. First one is text based query, protein name, name of the author who has deposited the structure or any particular name of ligand of the interest that can be entered in the search box. The next method is sequence based query. In this option, protein sequence can be provided in the sequence search option. And last one is the searching by using PDB ID. One can search a structure in PDB by using 4-letter code or keywords related to its annotation. PDB ID is a 4-character unique identifier of every protein which deposited in Protein Data Bank. Each structure published in PDB is assigned a 4-character alphanumeric identifier at the time of deposition. The first 4 character is numeral in the range 1 to 9, while the last 3 character can be either numeral in the range of 1 to 9 or letters in the range of A to Z. If the PDB ID of interested protein is known, it is the most direct way to retrieve it from the database. The data in PDB is usually stored in 3 different file formats: PDB file format, MMCIF file format and PDBML. The Protein Data Bank file format is a textual file format describing the 3-dimensional structure of molecules held in the Protein Data Bank. It can be read in Notepad in case of Windows or gedit in case of Linux. .pdb file are read to use by ResMol to display the molecule. A typical PDB file describing a protein consists of hundreds to thousands of lines like presented in this figure. The figure consists the header, title and author record. The header, title and author record provide information about the researchers who defined the structure. Numerous other types of records are available to provide other type of information, like remark records can contain free form annotation, but they also accommodate standardized information. For example, the remark 350 biomart record describe how to compute the coordinates of the experimentally observed multimer from those of the explicitly specified ones of a single repeating unit. Sequence record give the sequence of the 3 peptide chains named A, B and C, which are very short in these example, but usually consist multiple lines. Atom records describe the coordinates of the atom that are part of protein. For example, the first atom line above describe the alpha N atom of first residue of peptide chain A, which is a proline residue. The first 3 floating point numbers are its x, y and z coordinates and are in units of Armstrong. The next 3 columns are the occupancy, temperature factor and the element name respectively. Heteroatom records describe coordinates of heteroatoms, that is those atoms which are not part of protein molecule. The protein data bank markup language, that is the PDB-ML, provides representation of PDB data in XML format. The description of these format is provided in XML schema of the PDB Exchange Data Dictionary. This schema is produced by direct translation of PDBX or MMCIF Exchange Data Dictionary. Other data dictionary used by PDB have been electronically translated into XML or XSD schemas. MMCIF is the acronym for Macromolecular Crystallographic Information File. MMCIF is based on subset of the syntax rule for the self-defining text archive star file. A dictionary description language, that is a DDL, defines the structure of MMCIF dictionary. Dictionaries provide the metadata which define the content of MMCIF data file. MMCIF data file, dictionaries and DDL all are expressed in common syntax. How you can access this important database is for this: just open the browser and search for PDB in the search area and simply click it. The first website, that is a rcsb.org, is the website for the protein data bank. Just click on it. As you can see on your computer screen, this is how protein data bank homepage look like. This page shows the general information about the PDB database. It also tells about the molecule of month, which means this molecule is searched many times in the month of September. It also shows latest structure available on the PDB, and this section shows about the news regarding the PDB database. And so this is the basic home page of PDB. Let's try to explore the database in detail. There is a search box in the top of this page. Here you can type anything you want to search. We can find our protein through name or protein ID. Let's assume that you are interested in serine protease enzyme. Write down the name of serine protease in search box and just click on the search button. Search will begin, and after a few seconds all the structure related to your query protein will display on the screen. This is very important point, that for one particular protein you can get multiple structure. All of these structure related to the query protein. On the left side there is a breakdown of the result, where you will have an idea that how many structure are related to your query which belongs to human, mus musculus, severe acute respiratory syndrome coronavirus 2, synthetic construct, Bos taurus, E. coli and so on. This breakdown is with reference to the organism in the protein data bank. Let's assume that you are interested in 4-PRO. To get more idea about this protein, simply click on this PDB ID 4PRO. This will directly land on to the next page, which will be specific to your selected protein. Let us see what kind of information we actually got for our query protein. On the top there is a certain tab like tab. First one is the structural summary, 3D views, annotation, experiment, sequence. I will explain one by one. First is the structure summary. These tabs show you complete structural summary of your query protein. Here this first 4-letter code, that is a 4PRO, is a PDB ID. Then complete name of protein, DOI number assigned to your protein, classification is serine protease, organism is Lysobacter enzymogenes, and expression system of your protein is E. coli. Mutation, there is a no mutation in your protein, query protein. Deposit date of query protein is 1st October 1998 and the release date is 18 May 1999. Then it provides name of author who have submitted the protein to the PDB, and then this is the experimental data. Experimental data section shows that experimental method used is X-ray diffraction. Next section is literature section. Here it enlists those publications in which this protein has been mentioned. Next is macromolecule, which is the main protein. You can also search this article by clicking in the given link. Now move on to the second tab, that is a 3D view. Here it shows the 3D structure of your query protein. Just place the mouse and move its wheel on the structure. You can zoom in and zoom out of the structure. In the image, red dot shows the water molecule and green and purple ribbon like structure is protein subunit. Now the next tab is annotation. This section shows protein annotation and gene annotation and classification. This is the domain annotation, and this section also shows the gene annotation. Next tab is experiment. If you click here, this tab will show all experimental data that was used to determine the structure of the query protein. Here the technique used are X-ray diffraction, and next tab is a sequence. Here you get FASTA file sequence of your query protein, and lots of information are given here like sequence in PDB, sequence in UniProt database, secondary structure of the protein, etc. And these graphs show the complete information of the particular protein. You can download the FASTA sequence of your query protein by clicking on the download file tab. Next step is genome. It will give you the location of your protein in genome or the chromosome. Last tab is version, which shows its upgradation, means when the query protein was initially released in the PDB and when it was updated again. Your query protein was first released in 1999. After that it was updated in 2008 to 2011, 2020, 2023, and it was denoted by version 1.1, 1.2, 1.3, 1.4 respectively. By this way you can get so many important information about your query protein. So this is all about you can search about particular protein and how you can get various important features of your protein in PDB. Let us take another example of human hemoglobin protein and have a quick recap of all search strategy in RCSB database. Let us search via PDB ID, type 4HHB in the search box and simply click it. In structure summary, this tab shows complete structural summary of hemoglobin protein. Here this first 4-letter code, that is 4HHB, is PDB ID. Then complete name of the query protein is the crystal structure of human deoxyhemoglobin. Then the DOI number of this protein assigned by the PDB. Next is the classification of protein is oxygen transport, and organism is Homo sapien. Mutation, this protein does not show any mutation, and the date, this protein was deposited on 1984 March and release on 17 July 1984. Name of the authors who submitted this protein to the PDB, and this section shows the all experimental information of the hemoglobin protein. Experimental method used to get this protein is X-ray diffraction. Resolution is 1.7 angstrom. The resolution is a measure of the level of detail present in diffraction pattern and the level of detail that will be seen when the electron density map is calculated. High resolution of structure with resolution value 1 angstrom or more are highly ordered, and it is easy to see every atom in the electron density map. Then R value of this protein is 0.135. What is this R value? R value is the measure of quality of the atomic model obtained from the crystallographic data. When solving the structure of protein, the researcher first build an atomic model and then calculate a simulated diffraction pattern based on the model. In 3D view tab shows the 3D structure of hemoglobin protein and its sequence. In the right side of this page shows various information about this protein, like its structure, component of the protein like polymer, ligand, water molecule in the protein and ion. Next tab is density. Quality assessment tab shows validation report of this hemoglobin protein. Assembly symmetry of this protein is enabled. In export model option, one can export the model in MMCIF format. In annotation tab of this protein, following are the present annotation of the hemoglobin protein. First one is the domain annotation in scope or scope E classification. Second is the domain annotation in scope 2 classification, domain annotation in encode classification, domain annotation in cath data, protein family annotation, gene product annotation. In the sequence tab of this protein, there is a graph which shows the information about the sequence of this protein, its chain, binding site, metal coordination, etc. It also shows this protein in another database like cath database, scope database and encode database. In genome tab shows that this protein is located in chromosome number 16. The version tab of these proteins shows that this protein is updated many times and its latest version is 4.1, which is updated on 15 March 2023. Changes made a PDB entry after its initial release are considered to be either major or minor. The latest minor version of the each version is available as a download file. So, this is all about how you can search about particular protein and how you can get various important feature of your protein in protein data bank. With these lecture, it is clear that PDB is very important database when it comes to the area of structural biology. Structures in PDB have wide application. They can be used for various studies including identification of new protein, structure through in silico approaches, or can be used for protein nucleic acid interaction studies. With these, I conclude my lecture. Thank you.